Hi, my name is Zoey Armstrong, and I'm going to be presenting today about my project. It's called Evaluating the Effectiveness of Species Distribution Modeling for the Pacific Golden Chanterelle. Here's a short About Me page. Feel free to pause it and check it out. But I'd like to get into some background first. So a herbarium is a collection of preserved plant specimens and associated data. And on screen you can see an example of that. There's a preserved plant as well as some information in the bottom right of that image. In this case, I'm only interested in the location and the locality information for my project. But herbariums don't just collect preserved plant specimens, they also collect fungi and mushroom specimens. The largest aggregator of these is the microportal, which is an online website that searches through hundreds of institutions, and it collectively houses over 6.4 million specimens. So this is a large wealth of information that has not been studied in much detail. So my project aims to fix that by creating several statistical models using this herbarium data, assessing model quality with an independent data set, accounting for sampling bias with a weighting scheme, and trying to fill this gap in the literature. Well, there have been several studies looking at species distribution modeling of fungi. None have used an independent data set to validate it. So that's what I'm hoping to accomplish here. To do that, I'm going to use the Chanterelle formosus as my target species, also known as the Pacific Golden Chanterelle. This is chosen for a couple reasons. One, it's in a location with varied topography. Two, it has a lot of records, which is important for the modeling software to be accurate. And thirdly, it has a cultural significance in the region. As a forageable mushroom, it is very economically important, so understanding the distributions of it is important for that region. Here you can see what the herbarium observations look like. There's 244 throughout the state, and we can use these points to create a species distribution model similar to this example. As you can see, we have species occurrence data and environmental predictor variables. The occurrence data can extract the environmental data, and then this is run through a regression software or a regression package where you find out the contributions of each predictor variable and output a suitability map from that. For my project, I used these as my environmental variables, the most important of which ended up being pH, as well as nitrogen, cation exchange capacity, and canopy cover. DEM aspect and slope also were found to be important. But before I get to the results of this, I wanted to discuss spatial bias. So these points were sampled in a completely unstructured way, and as such, they could have sampling bias and spatial bias. So in order to account for that, I used what's known as an effort variable by looking at different things such as population density, distance to roads, terrain ruggedness. We can find out areas where it's easier to sample versus where it's harder to sample. So in this case, the areas in red will be weighted more heavily in the model than the areas in green, which are easier to get to. This should hopefully produce better results. And we can see an example of that here. This is two general linear regression models, one with the effort variable and one without it. The one with the effort variable shows that there's less predicted habitat in the lowland areas that might have predictions there, but just because they're easy to sample. So yeah, this is great, but we want to then validate this information. To do that, I chose citizen science data. The platform of choice was iNaturalist, where users can go out and record observations that are given a locality information. That's what this looks like. I had 259 observations throughout the study area, and this independent data set can then be used to run statistical comparisons. So the statistical tests I plan to run are the true scale statistic, the area under curve statistic, and the McNemar test. The McNemar test is used for comparison between different models to test which is performing best in that sense. And the true scale statistic and area under curve are used to test individual model performance. I haven't had the chance to do that yet, but I'm looking forward to finishing it this semester. So thank you very much for listening to my presentation. Yeah.